to say a couple of things that actually Bob brought up. One is that we represent 4 individuals among a very large number of individuals here at the Mind Institute and elsewhere on the campus who are involved in Fragile X research. I'd say probably there's somewhere in the neighborhood of 24 to 30 or so investigators, all of whom are working on this, and we're here basically representing all of this work by all of these investigators. I should also point out that although we work on Fragile X, we think of Fragile X as a window into neurodevelopmental disorders such as autism, so that because the roots or the origins of autism are varied and many, we feel that studying specific neurogenetic disorders is very important to help us understand the mechanism. So this is one such disorder, but there are many. This happens to be quite convenient because of the number of individuals and because we know so much about the model. It's one of those disorders that has a very high association with autism. So our real goal, the grand goal of all of this, is really not just this syndrome, it's to understand the broader mechanisms that are undergoing with neurodegenerative disorders so we can develop targeted treatments. Now, in terms of this syndrome, it's one of about 18 or 19 disorders that are known now that all have a particular type of genetic mutation, and that is an enlargement of a specific repeat of DNA sequence. In this case, CGG repeated many times, so CGG, 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 and so on. Myotonic dystrophy is another one. Huntington's disease is another one. In that case, it's a CAG repeat expansion. And interestingly, essentially all of these 18 or 20 or so disorders that have these expansions have something to do with the CNS, something to do with the nervous system. That's a very interesting observation we really don't understand at this point. So most individuals in the general population have fewer than about 45 repeats. This 55 here, between 45 and 55, is a gray zone where we think there may be some involvement, but this is just emerging. Individuals who have expansions of about 55 to 200 repeats were initially important because their children could have a much larger expansion over 200 repeats and therefore develop the neurodevelopmental disorder Fragile X syndrome, which is the most common inherited form of cognitive impairment. So until fairly recently, the importance of the premutation was that the gene is unstable and the mother could have a child who has a full mutation disorder. The full mutation, we know generally, once the gene gets that large, the mutation gets covered with methyl groups, and that causes silencing of the gene. And so the problem with Fragile X syndrome is that the gene is shut off, so you make no RNA from the gene, and therefore you have no protein. And so the Fragile X syndrome is due to the absence of a protein. Now, I want to tell you a little bit about a model for the next 4 or 5 slides of the principal model that we work with now for Fragile X syndrome. The reason for doing this is because Rondi will talk about this later in terms of targeted treatments. So our other goal in studying this disorder and other developmental disorders is that they provide us with better opportunities for targeted therapeutic intervention. This has been a real problem not only in the neurodevelopmental disorders but in the neurodegenerative disorders, Parkinson's disease, Alzheimer's disease. We really don't have good targeted treatments that are based on the mechanism of disease formation. So we're trying to understand that kind of approach to treatment by understanding the mechanisms of these diseases. Now, in the case of Fragile X, I want to start with a very simple, this is the level I understand it, a simple model of how the synapse works, the connection between 2 neurons, which provides a source of communication. So in response to stimulation of an axon from one nerve, in this case, there is a neurotransmitter, glutamate, that's released from one side of the synapse across to these receptors, the so-called AMPA receptors, based on the fact that there are drugs that we know that can block particular types of receptors or activate them. So once the glutamate is released in response to a signal from this nerve, it runs across, it activates these channels, and metal ions go in through this through this spine on this neuron, and then you get a signal going back to the nerve. So this is the way things communicate. Now, we know a little bit more about it, and this is really important for understanding the way we would treat these individuals, and Rondi will talk about the treatment. So the receptors themselves normally cycle. They're on the surface where they're active, and they go inside where they're inactive, and they're either destroyed if they're defective or they move back out again. So this cycle normally takes place, and the amount that's in here determines how much activity this nerve has. So this is a very important regulatory mechanism. Now, this other receptor, let me just go back, this is called a metabotropic receptor because it doesn't actually act as a channel, it just acts as a signaling device. If you bind glutamate to this, it signals internally to the cell. So it's a different kind of receptor. So if you activate that receptor, it acts a little bit like a brake. So basically what happens is when you signal this thing, it ends up slowing down. Actually, it ends up causing a little bit more internalization, therefore slowing down or reducing the activity of this synapse. So it tends to help pull things in. If you stimulate for a longer period of time, you get actual translation of the RNA for various genes and more protein synthesis, and that actually pulls these in more, so it actually downregulates this even more. It's a little bit like a brake on a car. So what we found out, we meaning the field of Fragile X research, was that one of the proteins synthesized in here is FMRP. This is the Fragile X protein, and FMRP actually puts the brakes on this. So it's like a second braking mechanism for the brake, so you don't brake too much. Because if you didn't have any kind of control on the braking mechanism, you'd pull all this in. So the cell's very finely tuned to have just the right amount of activity. Now, the problem is in Fragile X syndrome is that second brake is not there. So if you remove Fragile X, all of this protein synthesis pulls these receptors in more and more and more, and that weakens the synapse. So when you weaken the synapse, you affect the connectivity in the brain, and you really don't have any way of slowing that down. Okay, so the absence of this protein brake here and the excess pulling in of these receptors gives rise to Fragile X syndrome, and an obvious target at this point would be this mGlu-R receptor. So that if you could block its function, you would effectively reduce the amount of this going on, so you could get around the problem with the absence of the protein, and so provide a little bit more receptor function on the surface. So this is a way of getting around the problem. You're not replacing the protein, but you're recognizing what the protein does. And this is the essence of targeted therapeutic intervention. If you understand the mechanism, then you can develop better drugs for targeting. Now, one of the things that we've been working on very, very hard is to ask, well, how much protein do people have? Now, it's been known that if you look, these are males and females, and we now have a quantitative test that was developed by Christine Iwahashi in the lab for actually measuring the amount of protein that an individual has. You can do this in brain if you have brain tissue, or you can do this in blood, peripheral blood. If you look at individuals with full mutation, there's very little protein being produced. There's a slight variation here, but mostly it's very low. In premutation individuals, it's down just a little bit, but in normal individuals, and this is what we found, which is quite shocking, is there's a broad variation in protein levels in normal individuals. Now, this wasn't known until quite recently with the advent of these tests, and what this suggests is there may be issues involving how that a protein affects cognitive ability even in the normal population range. And people here such as David Hessel and Andrea Schneider and others are actually looking at this question. In females, you don't see this kind of drop because females have a second X chromosome, and usually you're getting the average of the one that's shut off and the one that's not shut off. So we're now starting to see a lot of information about who has how much protein. And you might say, well, in a normal range, somebody who has a very small amount of protein may be more susceptible to other kinds of illnesses or cognitive problems because even though they don't have an expanded X chromosome, they may have problems with cognition. Now, in this regard, it's very interesting. A recent paper by Fatemi et al. In 2010 simply looked at the brains of individuals with major psychiatric disorders, and in a real shocker to pretty much everybody, found that when you look at three major disorders, bipolar disorder, clinical depression, and schizophrenia, that in all three cases, this is approximately 15 brains, this is the average result for 15 brains, that the amount of fMRP, this protein, is down compared to age-matched controls. Now, we don't know whether this is cause or effect, but either way, it's very important because it suggests that some of the cognitive decline that you see in some of these major psychiatric disorders may in fact be due to the loss of this protein, which is important for cognition. Now, if it's an effect, that's really important, and we have to ask, why does a psychiatric disorder give rise to reduced fMRP? If it's a cause, then it may be that those individuals in the normal population have low levels to begin with, may be at greater risk. So now we can assess the idea of risk. So this sort of issue is very, very important in thinking about the broader context of this protein. Now, I want to make a point that I made at the outset, and that is there are a lot of proteins in the synapse, and somewhere in here is fMRP, but there are a lot of proteins that talk to one another. And if you look, this is a very, very simple schematic. Here is Rett syndrome, MECP2. It's a very simple schematic. There are much more complex ones I could have put up of proteins talking to one another. And if you look at these proteins, various of these proteins actually, when mutated, give rise to other neurodevelopmental disorders, and many of which are associated with high levels of autism. So in understanding the pathways, not just the individual proteins, we may have a broader understanding of how neurodevelopmental disorders take place. Here's an example of a very interesting study done a few years ago where they simply looked at over a million patient records, 161 disorders, and they looked for associations, the type of associations you wouldn't expect and you couldn't see if you didn't use so many records. And at the center of this universe is autism. And they asked, how can we relate autism, the fact that an individual has autism, with something else they may also have? So a comorbidity. If they have autism and seizures or autism and something else, can we make some sense out of associations? This is very powerful for those of us who are thinking about mechanism, because if we say somebody has autism and they have a particular type of disorder also, we may understand one through study of the other. So somewhere upside down here is Fragile X, which is not surprising. But interestingly, if you look at autism, you'll notice that they see the size of the circle represents the strength of the association. That you see depression here, you see schizophrenia here, and you see bipolar disorder. Now, what you can think about, and going back to that slide where the low fMRP levels, you can think, wow, maybe these sorts of things are reflecting pathway commonalities that may give us some insight how to treat autism if we understand what schizophrenia is and vice versa. So this also points out something I want to stress, and that is we're looking at the studies of FXTAS, which is a neurodegenerative disorder that I'll mention in a minute. We're looking at a neurodegenerative disorder, and when our consortium, entry consortium was first funded, that's what we thought we were looking at. But what we found when we started studying this disorder is we kept going back and back and back in time, and now what we know is this is a disorder, a neurodegenerative disorder that hits in the 50s and 60s that really starts out at or around the time of birth. So now we've discovered that this is really a neurodevelopmental disorder and that the degeneration is a late aspect of this. And we would never have found that if we hadn't been able to study a broad range of symptoms, a broad range of ages, and try to connect those dots. So I am very interested in the concept of using such disorders like schizophrenia, bipolar disorder, and linking them to neurodevelopmental disorders, and we can understand more about both. So I want to talk a little bit about FXTAS. Fragile X-associated Tremor Ataxia syndrome was a disorder that was discovered about 10 years ago. It's a neurodegenerative disorder. It hits generally men, but also women in their 50s, 60s, or 70s. And I'm proud to say it was discovered by a developmental pediatrician here at the Mind Institute, Rondi. And it was discovered by her because she took the time to listen to the families who were bringing their children in with Fragile X syndrome, and the moms were complaining about their fathers, falling down, having a tremor, they can't write anymore, they were losing memory, and she put that together and made the connection with the gene. Now, most adult neurologists wouldn't have made that connection because they've never heard of Fragile X syndrome, and most Fragile X syndrome pediatricians really aren't talking about grandfathers and what's going on with those guys. So this connection was critical. It was a two-generation disconnect, but when you made that connection, it allowed us to understand that, in fact, the degenerative disorder really was the end stage of a developmental disorder. So what we have here in the premutations is, in fact, this gene is producing way too much RNA. This was a discovery made by Dr. Flora Tassone, another faculty member here at the Mind Institute. She made this observation in the late 90s and first published this in 2000. So it was a really good way of putting into context what Rondi and others clinically had been seeing in these young children who were carriers, that is, premutation carriers, because we now knew that there was a biochemical abnormality associated with the carrier state. And I'll mention in a slide or two coming up that, in fact, what we think is this excess CGG repeat is actually binding proteins abnormally, and this abnormality leads to problems. I want to show you part of the tremor in this individual here, which was the first individual.
It's okay if you miss, it's okay.
This was the first individual ever discovered with Fragile X syndrome, with FXTAS, and this was an individual observed by Rondi. You can see the problem with the tremor. At rest, he has no tremor, but when he's asked to do something, this is an intention tremor as opposed to a Parkinson-type tremor.
That's okay.
He retired as an electrician, which is a very important thing for somebody with the tremor to do. Okay, so what I want to show you, so what we also see is a loss of brain tissue in these individuals, so the brain is really shrinking. And the central finding in this disorder is that in the nuclei of neurons, these are neurons from the cortex, that you see these particles here. These are inclusions, and those are very, very important indicators of this disorder. We also know that there is mitochondrial dysfunction in FXTAS. This is a very recent finding by Cecilia Giolivi here at UC Davis, and also a member of this broader consortium. We see decreased oxygen uptake rates and uncoupling between electron transport in these mitochondria and synthesis of ATP. I won't go into this in detail. The important issue here is that if you don't have good mitochondrial function, cellular function itself is really impaired. And so the brain requires a lot of energy, and we think that the fact that these mitochondria are dysfunctional may give rise to some of the problems that we're actually seeing clinically. Now, Chantal Sallier and Nicolas Charlet in the last year have been looking at what proteins actually bind to the CGG repeat, and we now know a bunch of these, but very excitingly, two proteins called DROSHA and DGCR8, it's a very cryptic name, I'll explain in a minute, are very important, we think, for the initiation of this disorder. Now, what DROSHA does, and these two, this is a very complex slide, but I'll explain that when these two guys get together and bind on the CGG repeat, they basically pull themselves out of solution so they don't work anymore. And these two proteins are responsible for producing a huge range of thousands of different RNA molecules in the cells. And here they've just measured two of the many hundreds of RNAs that are down-regulated in FXTAS compared to controls. So we now have something where we're looking at the change in landscape of RNAs. DGCR8 stands for DiGeorge syndrome critical region 8, which is also a little bit abstruse, but it's related to another neurodevelopmental disorder studied here at the Mind Institute by Tony Simon, which is DiGeorge syndrome, or 22q deletion syndrome. That protein is one of the proteins that's missing in that syndrome as well. So now we have another link to another neurodevelopmental syndrome. Now, I just want to go from the neurodegeneration back to the neurodevelopment in the last few slides and point out that what started off as our study of a neurodegenerative disorder has again taught us about neurodevelopment. This is work that Isaac Pesa, another Mind Institute investigator, and Yusui Chen, a postdoc in his lab, found when they looked at neurons from day 1 mice. Now this is way back at the start of postnatal development of the mice, and what they found were the dendrites, the dendritic tree of these neurons in those mice were abnormal. So all the way back to day 1, we now know that there are abnormalities and in the growth and development of these neurons. And Steve Nochter, another Mind Institute researcher, with Robert Berman, another Mind Institute researcher, all working as part of this collaboration, found that the number of embryonic precursor cells was also decreased. So this is very, very important because it tells us that there are very early neurodevelopmental events that eventually would lead to a neurodegenerative syndrome. So this is just an ending. This is just an example of the spectrum of premutation involvement. We didn't really used to think there was really much of anything going on. Now we know there are neurodevelopmental problems, there's psychiatric involvement, there's endocrine dysfunction giving rise to the leading single gene form of premature ovarian failure or primary ovarian insufficiency, immune dysregulation, neurological problems of the original observations, and this disorder FXTAS. So I'll stop. This isn't done alone. This is a partial list of collaborators, many of whom are at the Mind Institute and some of whom are in other universities and other cities. Thank you.
Okay, I think we do want to save some time for questions at the end. So if I kind of flip ahead, it means I'm skipping, and I'll try to do some of that so that my next 2 colleagues can talk as well. So I think Paul beautifully set up my portion of our team talk here, because I really am, as Bob mentioned, really interested in neurodevelopmental processes, both in typical and atypical development. And so what I'm going to talk to you today is about some research that I've done, sort of an amalgamation of many different research projects that we've done that focus on Fragile X syndrome, but also he also set it up nicely because we're looking at the entire spectrum of Fragile X disorders, so both the full mutation and the premutation. And I'm going to take one specific example. My lab, in collaboration with so many of my colleagues here at the Mind Institute, has been looking at many different different aspects of these disorders. But I'm going to take one example, and I'm going to show you, hopefully, convincingly, how looking at just one aspect of cognition, the Fragile X has been really an exquisite model for us to understand neural development. So one of the research program questions that I've been focused on is, are there specific neural pathways that are exquisitely sensitive to fMRP? And again, you know, Paul has already set up for you what fMRP, or the FMR1 protein, is. And what we've been looking for in our work is, can we see evidence of a dose response in these brain systems to FMR1 gene expression? So we're looking at the brain development and expression, and we're seeing, are there certain areas of the brain that are more sensitive to FMR1 gene expression than others? And if so, are the pathways that we should target, these the pathways, in fact, that we should target for treatment and intervention? And so let me just sort of preface this premise by talking about these pathways of the brain that I'm, in fact, referring to. So in both Fragile X syndrome and autism, there's been something that's been deemed or purported a dorsal stream deficit. It has to do with visual information that comes into the brain through the retina, makes a Waylay station at the LGN, and goes back to primary visual cortex, then takes one of 2 pathways, either through the sort of dorsally through the parietal area of the brain, or ventrally down through the temporal area of the brain. And when the visual information takes these different pathways, it's different types of information that are handled by different parts of the brain. In this parietal area of the brain, it's handling information more related to spatial relations, motion information, visually guided reaching. So visual stimuli that have to do with motion, space, and time. The ventral pathway is more form identification and object representation. And as I mentioned, in both disorders, Fragile X and autism, there has been a purported dorsal stream deficit, meaning that if you look at the 2 types of visual information processing, there's going to be more deficits found in information processed in this area of the brain than information processed in this area of the brain. And connected to that, there's also this superior inferior parietal area of the brain, which is really squarely within the dorsal stream, that is associated with number and magnitude. And I'm going to show you just sort of a few research programs that have shown that in both sort of domains, both the low-level visual information that's handled by the parietal lobe and number and magnitude that's handled by the parietal lobe, we see very specific problems in Fragile X spectrum of disorders. So in fact, we've seen evidence of a dorsal stream or parietal cortex deficits in both neuroimaging studies and infrared eye tracking studies in work done in my lab in collaboration with others. So in the neuroimaging studies, we've done work, for example, these are just examples and just sort of snapshots, so there's more examples of these if I had more time. But so for example, girls with a Fragile X full mutation, we've seen problems in parietal cortex functioning. Adults with the Fragile X premutation, we've seen problems in parietal cortex functioning. In our infrared eye tracking studies, in infants and toddlers with Fragile X full mutation, we've seen evidence of dorsal stream or parietal function problems. And preliminarily, and I'll show you this sort of teaser data, we see some infants and toddlers with the premutation also have these problems. So first, imaging females with the full mutation. Almost all functional MRI in Fragile X syndromes that has been done with that have been done in the full mutation has been with females. Well, that's for obvious reasons. Girls with Fragile X have that extra X chromosome or a higher functioning in most cases than boys with Fragile X. They're easier to get into the scanner. Past work that I've done, actually dating prior to me arriving at UC Davis, focused on arithmetic processing in females with Fragile X syndrome. And this was pertinent to look at because in the phenotype of girls with Fragile X, you see a pattern of relative weakness in arithmetic, digit span, and block design, all these kinds of things that are handled by the parietal area of the brain. They're significantly worse on these 4 symptoms of Gershman syndrome, so dyscalculia, dysgraphia, finger agnosia, etc. So we know that they have in their phenotype problems with math, space, reasoning about space and time. This is just an image to show you the kinds of things that we do to get children with Fragile X syndrome into the magnet. First, we use a MRI simulator, or what we call the mock scanner, and we convince them that this is something they want to do. Not always easy. Sometimes we have a child who's literally rocking back and forth on their hands and knees on the mock scanner bed, and miraculously about a half hour later I have them laying on their back inside the MRI scanner. So we use all kinds of tricks and techniques to do that, but of course it's not every child with full mutation that can be scanned. So the task that we did a number of years ago, and this is collaboration with some of my colleagues at Stanford, including Alan Reese, whose lab I was in at the time, was a simple mental arithmetic task. So in the scanner, we asked the girls with Fragile X syndrome to look at these equations and press a button if the answer was correct. So there were both 2 and 3 operand equations. So you can see what the kinds of stimuli were, very simple arithmetic equations. This is their accuracy, just to show you how they did. So on the 2 operand task, where it's just 1 plus 3, for example, the girls with Fragile X were slightly, but not significantly worse than the controls. But on the 3 operand, they were significantly worse. So even those very simple arithmetic equations were difficult for the girls with Fragile X syndrome. Let me just quickly show you the imaging results. For on the 2 operand equations, on the top, you see these rendered brains. These are fake brains, and what looks like it's spray painted on this surface rendering is actually the brain activation that we observed. So for controls and girls with Fragile X, you saw sort of a similar kind of canonical pattern of activation, very characteristically bilateral parietal. That's what we would expect to see in math tasks, and also this parietal prefrontal network. You see the same thing in girls with Fragile X, although less robust, but generally the same. Now, when you switch to looking at the 3 operand equations, one thing I want to have you observe is that when you look at controls going from 2 operands to 3 operands, there's this very characteristic ramping up of activation. They're sort of gathering their neural resources to get the job done. Girls with Fragile X, however, don't do that. In fact, these look almost identical to one another. So just as behaviorally they're starting to fail at being able to gather their neural resources, so they don't show the same ramping up of activation as the typicals do. But this is probably the most interesting of the findings from that paper, which is that we observed a correlation between brain activation during those 3 operand trials and fMRP. So in girls with Fragile X, we have a variation of protein expression that we typically don't have in boys with Fragile X who are typically methylated, or the gene is typically turned off. And what we saw was the more protein that individual was expressing, the more activation we saw in these very sort of predictable regions, very specifically, some frontal and middle frontal and motor, but very specifically in the parietal lobe, supramarginal and angular gyrus. So we were very interested by that, and this really was our first, this was back in 2002, when I was still a postdoc, but this was my first inkling that there would be this dose response of expression, protein expression in this area of the brain. The other place that we've seen this dose response, and I'll kind of try to get to this a little bit quickly, is not in the full mutation, but in adults with the Fragile X premutation. Again, Paul nicely set this up for me, so you know what the premutation is already. It's those carriers of Fragile X syndrome, not expressing Fragile X at all, in fact, fairly high functioning most of the time. But we put them in the scanner, and we did a similar kind of task, but this time instead of doing arithmetic equations, all they had to do was do magnitude estimation. That is, tell us which side of the screen has more. So just press left if more is on the left, and right if more is on the right. And the very critical thing here is that that gets harder to do the closer together those two quantities are. So if I put up 7 and 9, that's much harder for you to tell me which one is more than if I put up 2 and 9. Okay, we call that the distance effect. And when we look at the distance effect, that is small versus large differences, we see that area of the brain, that parietal area of the brain, in both controls and premutation, but more so in the controls. But here's what's really interesting. So that's that between group analysis that I just showed you. Very interestingly low. We see a negative correlation with CGG repeat size, so that the higher the repeat size goes, the less activation we get in that area of the brain, which is that parietal area. And we see something similar with messenger RNA. So without more time to go further into this, this is just a little teaser to show you that both now in the full mutation and the premutation in neuroimaging research, we've shown that the FMR1 gene is actually, there's a dose response. So the more you're expressing of that gene, the more sort of normal canonical activation you get in the parietal area of the brain. The other sort of way that I've shown this is in a very different set of experiments. So this is a grant that I have to look at infants with Fragile X syndrome, the full mutation. And we've seen the same kind of dorsal stream or parietal deficits in these infants, and preliminarily in infants with the premutation. In these sets of experiments, we use an infrared eye tracker. I do not put the infants, so far, into the scanner and have them try to do things. That would be very difficult. But we put them in front of an eye tracker and show them certain kinds of stimuli, and just look at the way that their eyes are moving and what kinds of things they look at. This is sort of the larger study design, which I'll skip for a matter of time. But I can show you just that we have a whole lot of eye tracking paradigms that we do with the babies with Fragile X syndrome. We take lots of breaks, and the eye tracker that I use allows us to do that. But we try to get as many of these as we can. I'm just going to highlight one of those experiments today, which is this low-level visual processing experiment that we have already published on. This is a very good example of dorsal stream functioning, because in these stimuli we've pit the dorsal stream against the ventral stream. And the way that the baby looks at the screen tells us whether or not that area of the brain is functioning normally. So what we did is just very simple forced choice preferential looking paradigm. These are first order luminance defined bars. And so when this comes up on the eye tracker screen, which just looks like a computer monitor, we can tell where the baby is looking. And what we're seeing is if they look more to the side that has bars on it. These are all very high contrast bars, so you can see them very easily, but they go down to very low contrast, and we simply ask, does the baby look where the bars are? What we more specifically ask, we have these different contrast levels, is what their contrast sensitivity function is. That is, how much contrast do they need? How obvious do those bars have to be before they look to the correct side of the screen? And this is a page we took out of the psychophysics book. This vision scientists have been doing this for years and years. In this particular paper, we had 37 typically developing kids who were chronologically age matched to the 32 babies with Fragile X. These are showing those contrast sensitivity functions that I mentioned to you, so just showing you some sort of individual differences. And this is where we saw a difference on the second order motion stimuli. So when the bars are moving, like moving gradients, and they're made up of, they're texture defined, sorry, I haven't really defined that for you, but they're texture defined, that's where we saw a difference in infants with Fragile X syndrome. This is exactly the same data, showed a different way. So here's the babies with Fragile X in the dark bars, and they're the same, different but not significantly so, but significantly different on second order motion. Turns out that second order motion is really motion that has something to do with attentive tracking. Not only is it in the dorsal stream, but in fact to be able to see that kind of motion, you have to be able to use attention and track across space and time. So these things sort of all connect on some level, even though they're very different types. This is very different from math or magnitude estimation, but it's really using the same kinds of functions and areas of the brain. And so this is, in fact, this data is consistent with a dorsal stream deficit, but more specifically, an attentive tracking deficit. This is new findings on babies with the premutation, and it's very preliminary and very new. So I wouldn't, this isn't ready for a paper yet, but we're pursuing and trying to get more babies with the premutation. And the bottom line, the punchline is that that same difference on second order motion processing that we saw with babies with the full mutation, we're seeing with babies with the premutation. So in the blue bar, this is babies with the pre, this is babies with the full, and these are typically developing babies. And this is the contrast sensitivity. So we're seeing what we saw in the full mutation babies in the premutation babies, which I have to admit, I almost fell off my chair when I saw that data. It's not something I really expected, but it's really consistent with the message that Paul was talking about in his talk, which is that this is really a neurodevelopmental disorder. So it makes sense for us to be looking at children early in their development because this is something that clearly starts much earlier than when they're adults, and in fact much earlier than when they're older men, particularly those who are going to develop FXTAS. So that's just a summary. I think I'll just finish up with one more point, which is that we're now trying to use the knowledge we've gained to help the kids that we study. We're starting to pilot some game-based interventions that specifically target this parietal prefrontal pathway that I've been babbling on about so much, that we've shown to be FMRP sensitive in Fragile X syndrome and in the premutation. So we have this Track FX game that we have a lot of our families playing now, and my hope is that by playing this very specific kind of game-based intervention, that we'll actually be able to perhaps one day sort of shore up these neural pathways. It's important to do this early in development because, as we know, plasticity has a window to it. So that's something we're very excited about. My last slide is the acknowledgments, and this is only a very small subset of people in my lab and across the Fragile X team and the molecular genetics team that make this all possible. And of course, thank you to NIH who makes it possible, but most importantly to our families who really are my heroes every single day that I do this work. Okay, thanks.
This is my fancy title slide. I'm trying to save time. Which will give me a chance to talk, to set the stage. Basically what I wanted to start with is by making the connection between some of what Paul was talking about regarding the mGluR5 receptor and FMRP, which he nicely laid out, and make sure that you understand that the targeted treatments that he was starting to talk about and that Rondi will talk about in much more detail came about from a really deep understanding of the connection between FMRP and that receptor system. And that work led to a number of studies using the animal model of Fragile X, in general, the mice that have this gene knocked out, or fruit flies that have this gene knocked out. And in those animals who show a range of different behavioral, cognitive, and neuroanatomical deficits, the drugs that have been developed to normalize this FMRP mGluR5 connection have almost completely, in every test, normalized normalized functioning. So those drugs have not just improved functioning or improved neuroanatomy, but actually have completely normalized it, made it as normal as a mouse that does not have this gene knocked out. So that's something that's really important to emphasize. And so you can tell by what I'm saying that there's been a huge amount of progress in the development of this theory in the animal models. But the development of outcome measures that can be used in humans to determine whether those treatments are going to work in people with Fragile X, it has lagged very much behind the work on the neurobiology. So that's kind of where I'm focused. So in the lab, actually before I get to this, I'll make one other point, which is that because we're studying whether people with intellectual disabilities are improving related to treatment, we can't just ask them, do you feel better? You know, do you have less anxiety? Do you feel less fidgety or hyperactive? Because they really can't give us a reliable response. So we rely very heavily on parents to tell us whether their kids are improving, which is helpful, but as you can imagine, there are placebo effects that are very strong when you have parents reporting, biasing and other things. So we really want to have objective measures that we can use in combination with the parents' report. And that's basically what my lab is focusing on these days. One of the measures that we've focused on is called Prepulse Inhibition. And basically what it is a measure of sensorimotor gating. It's a way to index how the brain filters sensory information. And without going into a lot of detail, you can measure the startle response. You can see here is a fairly strong response in reaction to a loud sound. And when that loud sound is preceded by a softer tone, you get a much smaller response. So prepulse inhibition is how much this baseline response is inhibited by this tone. And in people with Fragile X, we've shown that they have very abnormal and reduced prepulse inhibition. So these bars here are female controls and male controls with nice prepulse inhibition, around 50, 60, 70%, and in the Fragile X groups, much impaired prepulse inhibition. And this really fits with their clinical picture because they are often overwhelmed by sensory input. They have trouble filtering it out. We've also been able to show that if you use this test at 2 points in time, it's very reliable. So at time 1 and at time 2, you get a very similar abnormal response in this measure. And that's useful because if we're going to use it as a potential outcome measure, we want to be sure that it's a stable trait in somebody before we look to see if it is improving. Another study that we published subsequently looked at people with Fragile X who have autism compared to people with idiopathic autism who do not have Fragile X. And you might be surprised to know that although many individuals with idiopathic autism also have unusual sensory responses, they actually were no different from typically developing kids as compared to individuals with Fragile X with autism who had this, again, impaired response. So we think that, excuse me, the mechanisms underlying sensory abnormalities, at least sensory filtering abnormalities, might be quite different in people with Fragile X autism and regular autism. I'm going to skip to save a little time. This measure, PPI, is now being used as an experimental outcome measure in at least 3 different clinical trials of these targeted treatments in Fragile X patients. One by Hoffman-LaRoche, another by Novartis, and a third by Seaside. These are just the titles of the studies that Rondi will be talking about more, but just to show you that we're making use of these outcome measures in some of the ongoing trials. Another big problem for individuals with Fragile X is anxiety. It's almost a pervasive problem in this condition, but nobody had really gone to determine how common is a formal anxiety disorder in this population. There were lots of self-reports and so on. And this is a paper by Lisa Cordero that's now in press looking at formal anxiety disorders in children with Fragile X. And you can see across the board some variability. The black part of the bar shows you what percentage of our population met full diagnostic criteria, and then the white section shows that many more people who actually had clinically significant anxiety symptoms but did not meet formal diagnostic criteria. And you can see specific phobia, very high rates, social phobia also high, and in females, really a similar picture, not a whole lot of big differences of note, but just very high rates. Many patients had multiple anxiety disorders, and overall, approximately 80% of the sample met criteria for at least one formal anxiety disorder. So a really, really big problem. We're kind of targeting the physiological basis of, in particular, social anxiety in Fragile X by using a protocol that's actually similar to the PPI one I was mentioning before, except in this case, we're looking at how emotional expressions can potentiate, not inhibit, but potentiate the startle reflex. And we know from animal studies and neuroanatomical studies that the amygdala is very involved in this startle potentiation, this adaptive response that gets us ready to deal with threats. And just we have some preliminary data that basically shows in our healthy boys and girls that both negative emotional expressions and positive expressions elicit a greater startle response than neutral ones. And this is basically telling us that our protocol is valid, that it is doing what it's supposed to do, and we're now beginning to look at how these measures play out in the Fragile X group. We also are doing eye tracking studies using the same technology that Susan mentioned to look at the issue of gaze avoidance. It's a big problem, as you know, in people who have autism, and also a real significant problem in the Fragile X population. So we have, we're looking at both where on the face individuals with Fragile X are looking compared to healthy controls, but we're also measuring the degree of pupillary dilation. So your pupil dilates and constricts depending on how much light is coming into your eye. But if you can control for the amount of luminance by preceding the image that you're interested in by sort of a scrambled up picture that has the same amount of light, you can measure how much dilation occurs in response to the image of interest. And we think that one reason that people with Fragile X have difficulty maintaining eye contact and engaging reciprocally is because their sympathetic nervous system is overreactive. And the pupillary response, or the pupil, also is changing in size depending on how aroused you are, how much your sympathetic nervous system is active. So here's just an example of a person with Fragile X in the eye tracking protocol, and you can see kind of where they're looking with the red dot. The picture of the patient down here. And you can see looking at the nose, looking at the mouth, a little bit at the eyes, but quite a bit less to the eye region than other places. And so the data basically bears that out, both for calm faces and happy faces and fearful faces. We see much more looking to the eye region in healthy controls and less so in patients with Fragile X, which fits the clinical picture with them. As far as pupil diameter goes, we don't see significant differences for calm faces, but as expected, for happy faces and for fearful faces, patients with Fragile X show this abnormal greater dilation of their pupil in response to these pictures of human faces. This is a reliable protocol, so if we do this test again at different points in time, you can see these scatter plots that just show very strong correlations between time 1 and time 2 on this protocol, so we know it's a stable trait. And it's really amazing if you look at these profile plots of visit 1 in blue and visit 2 in green, you even see almost an identical sort of trajectory of the pupillary response to these images. And you can see on the left, this is the typically developing group, and on the right, Fragile X group with this very sharp, sudden response. Again, just more data showing that it's stable across time. So moving on to some of the executive function deficits that are really problematic in people with Fragile X, attention, planning, organization, impulse control, all those things are big problems. So we've actually adapted or adopted, I should say, a test developed in Germany that taps into all kinds of aspects of executive function in a really kind of child-friendly way, as you can see here. It looks at alertness, distractibility, divided attention, flexibility, go, no go, which is impulse control. And we've done a study in collaboration with Liz Berry-Kravis at Rush University in Chicago. And what we found is that this protocol, at least some of these, wrong way, some of these are doable by Fragile X patients and are valid. And you can see here on the left the measures of the KITAP and what they're correlated with in terms of parent report of similar kinds of problems. So we don't have to spend any time going into detail, but the problems that are demonstrated here seem to map on fairly well with the problems that parents report. And then we won't spend too much time on this, but ICC is just a measure that tells you how reliable these measures are across time. Basically, you know, how easy, I should say, how feasible it is for these patients to do this test. We're also focusing a lot on parent report of behavioral problems. So when we go to the FDA to ask for approval for a drug that's being developed for treatment of Fragile X patients, we're not going to go to them and say, well, we want approval to measure PPI or measure whether PPI improves in patients with Fragile X. We really want a clinical measure. And so this was a meeting that was held at the NICHD where we had a consensus decision about the domains of abnormalities that really need to be focused on these clinical measures. And you can see what those are here. I think I'm going to skip this to give Rondi some time. There's a measure that's been chosen already by all of those pharmaceutical companies that are testing these drugs, called the Aberrant Behavior Checklist. It's a good measure. It's been used as an outcome measure in lots of autism clinical trials and in clinical trials of people with intellectual disabilities. And these companies have, for good reasons, gone ahead and just chosen that as an outcome measure. But we really don't know if it's reliable or valid in this particular population. So I'm heading up a multi-site study that's looking at the psychometric properties of this particular measure in the Fragile X population to see if we can actually recommend it as an appropriate measure in these clinical trials. And you can see the different sites that are participating in that. Our preliminary data suggests that the way that this measure is organized is really not a good fit for the Fragile X population. The items here under mood lability, they were all included in the original subscale that was developed by the authors. And you can see that all these ones in black are in the place that you'd kind of expect them to be. But some items that are in red here that originally were on a particular scale that the authors decided made sense actually don't fit in the Fragile X population. So we're actually thinking that we're going to have to reformulate this measure and recommend it a little bit to be used a little differently in the clinical trials. And I am going to skip unless you think there's time for me to, I don't know. How much time do you need, Rondi?
Well,
we're kind of going over, right? So yeah, I think I'll let you get going. So that's my lab. And now it's Rondi's.
I want to thank extended team members, and not even this picture includes all the people on our team. Let's see, here's Louise, here's Paul, but Flora's sitting in the audience. Here's many people on our clinical team. We don't have all the molecular team. People in Flora Tassoni's lab, people in Paul's lab are not really demonstrated here, but we have a great team of people. I want to point out that I do have some conflicts in the sense that I spent a lot of time talking drug companies into doing targeted treatment clinical trials here, and they pay for it, and I am very happy to work with many of these companies that have mGluR5 antagonists, GABA agonists, and different kinds of treatments that we think can really reverse Fragile X. There's a lot of treatments out there. You heard a little bit about mGluR5 antagonists. I'll tell you a little bit more. We've just recently completed a controlled trial on arbaclofen, which is a GABA B agonist. I will also talk about minocycline, which is a treatment that we're doing now. But the point here is there's going to be more than one intervention that will be helpful for these patients. I think to completely reverse intellectual disability and autism, it's probably going to take more than one med and very intensive educational interventions. But this is where we want to go. We want to cure Fragile X, we want to cure autism, we want to cure other neurodevelopmental disorders. And the exciting thing is that what single medication may work for one disorder, like an mGluR5 antagonist, which clearly works for Fragile X, there is now evidence that the mGluR5 antagonist will work for idiopathic autism too. In fact, Jackie Crawley was here a couple of weeks ago showing that an mGluR5 antagonist reversed autism in the TBTR autism animal model that she has. So we're very excited about some of the preliminary results in autism also. So just to review for you what Paul mentioned is this mGluR5 upregulation in Fragile X that FMRP normally puts the brakes on, but without this brake, this is upregulated. It leads from strong synaptic connections to weak ones, and that's the problem in Fragile X and probably many other disorders, is there are weak synaptic connections that could respond well to mGluR5 antagonists. But let me just say here that there are a lot of mGluR5 antagonists. We've looked at phenobom. This is the AFQ is the Novartis agent. This one is the Roche agent. This is under current targeted treatment trials right now here at the Mind Institute for adults with Fragile X syndrome. But arbaclofen works by a different mechanism, lowering the amount of glutamate here, which seems to be quite helpful to patients. But there are a variety of other medications. I'll talk about minocycline, which is lowering MMP9 protein levels that is strengthening synaptic connections and working quite well in young children. We tried CX516, which is an ampakine. It was not helpful in Fragile X, but there are more powerful ampakines out there. So the name of the game is we probably have to use a medication that hits the glutamate system to calm it down, another medication that maybe upregulates the GABA system, which is underactive in Fragile X, and probably in some types of autism and other neurodevelopmental disorders also. So there's more than one way to cure these disorders, and it probably will require a combination of treatments. Again, the mGluR5 antagonist, we tried phenobom. It was marvelously helpful, and then the company went bankrupt when their fluoxetine medication turned out not to be helpful for autism. So it went bankrupt before we could get more phenobom from them. But Roche is a very robust company. Novartis is also a very wealthy company, and Seaside Therapeutics, which is a much smaller company. I was on the phone with them yesterday, and they want to start at least phase 2 trials in Fragile X, hopefully before the end of the year, but it will probably be more likely to start beginning of next year. What we've seen anecdotally so far from these medications is very, very positive. Even though these are controlled trials, a whole group of parents has said, oh my gosh, how helpful these medications are. And their kids are talking more and having less behavioral problems, less anxiety, etc. Now, minocycline was first studied in the mouse. It normalized the synaptic connections. Here's the wild type mouse showing very strong mushroom-shaped synaptic connections. The Fragile X knockout mouse has a lot of thin, immature connections, also called Philippovia. And then when you added minocycline to the Fragile X mouse, it normalized the synaptic connections and improved some cognitive and behavioral tasks. We did a survey of minocycline in about 50 patients with Fragile X. Agustini Utari from Indonesia was here with us for a year, a pediatrician from Indonesia, and she found a very positive response in this, survey of patients treated with minocycline. So now we've been funded to do a controlled trial in Fragile X patients, and we're just very close to finishing this trial. So we're looking forward to the analysis, and it will have many of David Hessel's targeted outcome measures for this minocycline treatment, so we'll see how well these measures work. Now, one of the problems of minocycline is it can cause graying of the teeth and sometimes some graying of the fingernails, but a lot of parents choose for better synaptic connections, even though there might be some graying of the teeth or fingernails. I want to talk about our baclofen, which is a GABA B agonist, about 15 times more potent than regular baclofen. Is actually the right isomer. And it has been very helpful in the Fragile X animal model. It's also been shown to be helpful in some of the autism animal models now. So even an open trial in autism patients without Fragile X was very positive. So now we're going to start a further controlled trial in Fragile X here at the Mind, and a controlled trial in individuals with autism without Fragile X at the Mind. And that will get going, we hope, before the end of the year. Just to show you how our baclofen works, it's a GABA B agonist, and here's the receptor here. When it stimulates the receptor, it actually lowers the amount of glutamate at the synapse, and that downregulates the mGlu-R5 system, lowers the amounts of excess proteins that are produced in Fragile X syndrome, and that helps with cognition and behavior. So the double-blind controlled trial that we did, and I'm going to show you some outcome measures of this. As you can see, they were on first randomized to either our baclofen or placebo, getting up to an optimal dose, staying on that optimal dose for about 4 weeks, then being washed out and going to the opposite treatment. All of this is blinded. Outcome measures were taken where these arrows are, and I just want to show you some results of that. We looked at patients between the ages of 6 and 40 with the Fragile X full mutation, Fragile X syndrome. Many of them had autism, others did not have autism. We looked at individuals that had significant irritability. We started with a very low dose, went up in the dose to an optimal level, and then kept them on that dose. Now, we did this in 63 patients. Some actually didn't complete the study. 89 did complete the study. So I'm going to show you some of the safety data. Very few side effects. They were mostly males, fewer females. I'm going to show you the data on the 54 that met the protocol of, actually carried through in the protocol. I'm going to show you the data on those with Fragile X and autism only. And I'm going to show you the data of the low sociability group. So these are subgroups within the Fragile X syndrome patients, and we think that these individuals actually had the best response. So the CGI is a clinical global impression scale, and the clinician who is blinded to what they're on talks to the family, see how the patient does in clinic, and makes a judgment as to whether they were very much improved, or minimally improved. Actually, the parents and the researcher looks at this. And the responders would be those that are either much improved or very much improved. And the light blue is the R-baclofen, the dark is placebo. As you can see, for the overall group, the 54 or so that completed the protocol, there were 35% that were responders, but 18% that were responders to placebo, and that wasn't a significant difference here. But when we look at those that just had autism, in addition to Fragile X, we found a much better group of responders. 44% responded to the R-baclofen versus 6% responding to placebo. So that's a very low placebo effect. So that was very positive, meaning that those with autism seem to respond the best. Those with low sociability, as measured by the ABC checklist, also that includes those with autism, but others that don't quite meet the criteria for autism. Again, we see almost 60% which were responders versus 19% on placebo, and that also was significantly different. When we look at other kinds of outcome measures, like the ABC social withdrawal scale, the Vineland socialization domain, and looking at responders either on the CGI combined with a 25% improvement on the sociability scale, there were far more significant number of responders versus placebo. So not everybody responds well to the medication. It could have to do with some molecular background gene subtyping or some of the circumstances in the family. And I think for each of these medications, we'll see a variability in response. Clearly with the R-baclofen, those with autism and those with low sociability responded the best. So now in the controlled trial, we're not only going to look at autism without Fragile X because they did so well on their open trial, but we're also going to look at individuals with low sociability, so trying to pull out that subgroup in Fragile X. In the future, we have plans for combining targeted treatment like R-baclofen, because we can start now as young as 5 years of age, so that's very exciting. We can start with young kids and combine it also, particularly those nonverbal ones, combining it with an intensive language intervention that Len Ambadudo has designed, but also think about combining it with other sorts of computer games to enhance learning. We have one called the CogMed program that Julie Schweitzer here at the Mind Institute has used a lot with ADHD, and we'll see if it works well with targeted treatments, R-baclofen. Flora Tassoni has a newborn screening study. We are identifying individuals with the premutation and the full mutation. We are finding that the premutation is much more common than we thought in the general population. It looks like it's anywhere, depending on what month you look at and how you combine the data, but it's around 120 to 150 in the general population has the premutation. The full mutation is more like 1 in 2,500 to 1 in 4,000, but the newborn screening data not only gives us babies that we can think about early intervention projects with, and thinking about combining it with intensive therapy, such as Sally Rogers' Early Start Denver model for autism could also be very helpful for Fragile X babies. And maybe we need to think about minocycline even earlier. So we're very excited about working with very young kids. We also have targeted treatments going in FXTAS2 to block glutamate toxicity, but I wanted to kind of get you excited about these proposals. We, in the next 6 months, will be starting about 6 more studies. So we really want to draw many more people with both Fragile X and also with autism without Fragile X into our targeted treatment studies, and we're very excited about maybe in the future starting targeted treatment studies with Rett syndrome, 22q deletion, and we're doing an assistive technology study now with nonverbal patients with Down syndrome, but I think we will really expand our targeted treatments, but with a very great focus on autism and also on Fragile X syndrome. This is a little bit of grain of the teeth that this little boy got, but he was a great responder to minocycline, and for his parents it was very worth that side effect. So I want to again thank the clinical team, thank our molecular team who are always thinking up new things to try in patients and testing them in animals and cellular models and iPSC cells. And it's really great to have a multidisciplinary team of people to do this type of research with. And it's also very exciting for the families when you can offer them great treatments that will make a difference for their children. And so we hope to cure these disorders, and I think we will.
I'll be crystal clear. I know you must have some questions. This is really, we're really in a new age of targeted treatments. So Flora, please go ahead. This is Dr. Portasoni, who's a molecular biologist here at the mine and runs the newborn screening and many other studies.
So I have a question for Susan. So I know that you were surprised when you tested those premutation with your tracking test. And it was kind of maybe a coincidence that you actually did that, or because you were, let's say, pushed. But
I didn't say that, you did.
But then, aren't you surprised to see that the deficit is kind of in the same direction that the Fragile X? I mean, when we think about like commonality across disorders, and then you had to match what you're testing and what the phenotype is, and ultimately with a cellular molecular abnormality or abnormal phenotype. But then, how do we reconcile the type of results that we get when we know that molecularly is different?
Right. No, that's a very good point. I mean, that's part of the surprise to those results. But the same thing would be true of the adult results with the neuroimaging. So it's in the same direction as well, right? So we don't know what's going on exactly, except that there is some sort of variation of function going on. And we know that there's some interplay between, I don't know what it is, elevated message, slightly lower protein at the higher end of the premutation, so that it isn't, I don't think, a different mechanism, at least where the cognitive neuroscience is involved, but it is a continuum. And so when I'm seeing this in the premutation, I'm sort of picking up something is rising, the signal is rising above some baseline, and it's because that signal is there in this continuum, and I expect it to be in the same direction. Or not, I don't know.
It's the room, don't worry about it.
It's the room, okay. I do expect it to be in the same direction, but I do expect to see this correlation, so this response that I was mentioning. Is that satisfactory, Flora?
Yeah, and also because, the study that Paul reported about these other neurodevelopmental disorders and other, that they show that actually fMRP is low, or at least is much lower, then you would expect maybe to make the same kind of connection there too. But then you think, like, schizophrenia, why they don't have, if it's true that there are like 24% of fMRP level that in a normal individual, then you would expect to see something about Fragile X in that population, but maybe not. Maybe because they always had fMRP during their life, and then something later in life happened, and he dropped fMRP, so they had that protein was present, so maybe when it was important to be there. But then if you test, I mean, I'm just confused about, how we're gonna make this connection when you talk about clinical phenotype and what you test for, and then how you report to really the normal molecular phenotype.
No, I think it's valid for you to be confused about that, and I think we're just we're just starting to understand that. But I think maybe part of the answer is going to be, and these guys might have an answer as well, that we have to sort of reframe our ideas about what those clinical phenotypes are. So we're sort of out of the era, in my opinion, that we're just going to look at the full mutation and that's what the phenotype is, because we're seeing these very subtle phenotypes in the pre as well. So it is a continuum, and this is a dose responsive. So our job now is to understand what the interplay is between gene expression, protein expression, messenger RNA, repeat size, other background gene effects, etc. Rondi wants the mic.
I was just going to say, we're looking at fMRP in blood. We're not looking at it routinely in brain. And I think in premutation, there could be pockets of fMRP deficit in the brain, not only in other disorders, but definitely in premutation too. And we're not even sure what happens through development. I mean, we think that fMRP is very important in early development, and it it could be regulated differently in babies versus adults versus aging people. So I think we have a lot more to learn about FMRP, even in the general population and all the things that regulate it.
Could be also like temporal problems, like, you know, a little deficit of FMRP at the very beginning might be more important than the fact that they have a high message level, and maybe, you know, with time, that would be more lethal than the FMRP that was lower to begin with. It's just that, I guess, we just don't know for sure.
That's exactly why we have to look at it in children, all the way through development.
So all I can say is this is a very important protein, not only for Fragile X syndrome, but for everybody. This is very important, related to how your brain develops and maintains itself, even in adulthood and even in aging. And there may be a lot of interesting things that happen in life that may impact expression of FMRP and cause subsequent problems in cognition. Like depression is a good example of something where there can be cognitive deficits associated with depression. Is that all related to the FMRP deficit? We would really like to know, and we would like to know if the deterioration in schizophrenia is related to FMRP. And could mGlu-R5 antagonists help a lot more individuals than just autism and Fragile X syndrome? Or could mGlu-R5 antagonists help premutation involvement? So we have a lot of work to do. Okay, come on, more questions, yeah.
This is for Paul. Could you elaborate, if you can, a little more on the significance of drosha and DGRC8 in this inclusion body? I think you started to address that a little bit in.
Okay, so what we found, and we is mainly the work of Chantal Cellier and Nicolas Charlotte in Strasbourg, but it's part of a collaborative team. What they found initially is when they went fishing for proteins that interact with that CGG repeat, they pulled out a bunch of proteins, and two of the proteins they pulled out were these proteins DGCR8 and drosha. Now, those two proteins work together as a team. With this protein DGCR8 binds to the RNA, and it holds drosha there. Now, the concept here is one that's been thought of in a number of these diseases, and that is if you enlarge the number of repeats, just the same way you would in, say, myotonic dystrophy or Huntington. Myotonic dystrophy is probably a better example because the repeat doesn't code for a protein. The same way in Fragile X gene, the CGG repeat doesn't code for a protein. It's just there as the RNA. So it's like a sponge that sucks up proteins in solution, and when it does that, then they can't go on about their business. So in the case of these two proteins, DGCR8 and drosha, if they get sucked up, then they can't do whatever else it is they're supposed to be doing. Now, the importance of this protein drosha, which basically comes from the name of this gene in the fly, fruit fly, is that it's important for processing a whole class of small RNAs called microRNAs. This one protein produces a precursor that then goes into the cytoplasm of the cell, and these little RNAs called microRNAs bind to other RNAs and regulate their expression. So there are thousands of these little RNA molecules floating around in the cell regulating the expression, the translation of other proteins, and all of them have to go through this thing called the microprocessor. It's this drosha DGCR8. If that's screwed up, then the processing and the production of all of these other RNAs is messed up as well, and therefore regulation of the cell. So it's important for us to ask, it's not that they're completely missing, it's that they're down a little bit, and so there's subtle other difficulties that are found throughout the cell. What's known is that there are at least 20 or 30 microRNAs that are known to be down in this case, and the question is how many and what's the consequence of that. So you can argue that this is an overwhelming problem if you have thousands of these RNAs that are down-regulating. But if you think about a targeted therapeutic approach where you simply disrupt the interaction between DGCR8 and the CGG, the problem hopefully would correct itself. So again, in thinking about targeted therapeutic interventions, one target would be to just simply disrupt that interaction. Everything else will take care of itself. That's the concept.
I just want to say a few things about premutation carriers. We study them a lot. The vast majority of premutation carriers who come to clinic, like the mothers of the kids with Fragile X syndrome that we see, are doing very well in life. We do see high rates of depression and anxiety, but they're very bright. In fact, I've always felt that IQ is better in premutation carriers compared to, you know, other controls that we see. So there's some good aspects, perhaps, of having too much messenger RNA, and many individuals are extremely successful. We see a lot of doctors and lawyers and ministers and jet pilots and legislators. So we don't mean to say that everybody with the premutation has developmental problems, but we see these problems in the ones that present to us clinically. And, you know, nobody really knows what percentage that is. It's probably overall about maybe 10% of the young boys with the premutation who might present with autism or an autism spectrum, and probably less than 5% of the girls with some of these neurodevelopmental problems. So we're very interested in what in the environment or perhaps in the background of the genes that could predispose them to autism. We have found that of the 7 premutation boys that we followed recently with seizures, 6 have developed autism. So we think that seizures is a good environmental stressor, if you will, that can push someone over to having more developmental problems. But we're very interested in other environmental and other factors in genetic background effects that could cause difficulties.
So David would like to answer a question. So if somebody could ask him one.
Bring it on. Come on. Don't we have a plant out there that can ask me a question?
Laura, ask David.
Dr. Hales.
Why do children with Fragile X have such high anxiety? What's your hypothesis?
That is a great question. Okay, so we know a little bit about that, but we need to learn more. So before I came here, I did some work showing that children with Fragile X, especially boys, but to some degree girls as well, have elevated cortisol, stress hormone. Whether that's a consequence of having the condition or contributory to the anxiety isn't really clear. That's one aspect. There's almost no question that the sympathetic nervous system is overly activated. That's been shown in our eye tracking studies and also in electrodermal studies where you measure the sweat response. It's very much exaggerated. So my guess is that it's not any one physiological component. It's probably limbic system overactivation, and it's borne out in all these different measures that we're using.
It also gets better with an mGlu-R5 antagonist, so clearly the upregulation of mGlu-R5 is, but the GABA system is downregulated, so some of the inhibitory systems, when they're not working, these other systems get really disinhibited, if you will. Okay, I think we're going to call it a night. Thank you all for coming out.
The UC Davis Mind Institute began in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, learning disabilities, and other brain disorders is helping children achieve their fullest potential. Please call or visit our website to find out more about current studies, our research team, and upcoming events.